consists of one of the ultimate ramifications of the main duct, ending in a number of cecal pouches or alveoli, which are tubular and somewhat convoluted. The minute ducts connected with the alveoli are narrow and lined with flattened cells. The alveoli are almost completely filled with secreting cells, so that scarcely any lumen is visible. In some animals, spindle-shaped cells occupy the center of the alveolus and are known as the centroacinar cells of Langerhans. These are prolongations of the terminal ducts. The true secreting cells which line the wall of the alveolus are very characteristic. They are columnar in shape and present two zones, an outer one, clear and finely striated, next to the basement membrane, and an inner granular one next to the lumen. In hardened specimens the outer zone stains deeply with various dyes, whereas the inner zone stains slightly. During activity, the granular zone gradually diminishes in size, and when exhausted is only seen as a small area next to the lumen. During the resting stages it gradually increases until it forms nearly 3/4 of the cell. In some of the secreting cells of the pancreas is a spherical mass, staining more easily than the rest of the cell. This is termed the paranucleus, and is believed to be an extension from the nucleus. The connective tissue between the alveoli presents in certain parts collections of cells, which are termed intra-alveolar cell islets, islands of Langerhans. The cells of these stain lightly with hematoxylin or carmine, and are more or less polyhedral in shape, forming a network in which ramify many capillaries. There are two main types of cells in the islets, distinguished as A cells and B cells according to the special staining reactions of the granules they contain. The cell islets have been supposed to produce the internal secretion of the pancreas, which is necessary for carbohydrate metabolism, but numerous researches have so far failed to elucidate their real function. The walls of the pancreatic duct are thin, consisting of two coats, an external fibrous and an internal mucus. The latter is smooth and furnished near its termination with a few scattered follicles. Vessels and nerves. The arteries of the pancreas are derived from the lyenal and the pancreatico-duodenal branches of the hepatic and superior mesenteric. Its veins open into the lyenal and superior mesenteric veins. Its lymphatics are described on page 711. Its nerves are filaments from the lyenal plexus. End of section 25.